Okay, thank you for this comment. Now let's proceed to a European project, disk-shaped point-of-care platform for infectious disease diagnosis, which was set up by Konstantinos Mitsakakis and his team with the goal of having a point-of-care diagnostic assays covering multiple febrile diseases in the field.
Thank you very much for the introduction, and I would also like to thank the organizers for the chance they gave me to present this European project with the fancy name Diskognosis. In fact, there could be, I guess, not a better introduction for the project aim and for the general background and the necessity of this project than the two previous talks, which expressed numbers and necessity and all this. I would simply like to add that in numbers from the World Health Organization 2010, 200 million cases have been reported before, 600,000 deaths, most of them, a large percentage of them, in Africa, and the cost about $10 or $12 billion per year. It was also reported by the previous speakers that in the same areas there are other similar diseases which have the same symptom with malaria, and that is fever. For example, dengue and chikungunya, which are viral diseases, as well as typhoid and pneumonia, which are bacterial diseases. So the problem is when a patient goes to these rural or even to the city hospitals, how can we distinguish which disease causes the fever? And in many cases, there's a false diagnosis, which leads, as it was mentioned before, to prescription of wrong medication. In addition, there is always the danger for spreading of these diseases, so we cannot say that they are localized geographically. And here I would like to quote a phrase that I heard last year in this meeting by Dr. Mouzin from Rollback Malaria. He said, "It is not enough anymore to tell a mother that her child has no malaria. She now requires to know what is the source of her child's fever." And I have to say that I was very happy to read this only 5 or 6 hours after the invitation I got from the European Commission to start negotiations for this project. So that day I got a double happiness. So it was also mentioned before, briefly, what are the state-of-the-art diagnostic tools for malaria. Rapid diagnostic tests, RDTs, which are based on immunodiagnostics. The advantage is they are cheap and easy to use. On the other hand, they can detect typically one pathogen per test. There is the microscope, which was mentioned that it has been used for years and years. The point is that it can only be used for malaria. And of course, there are more advanced molecular techniques like the PCR for DNA amplification. It offers, on one hand, high sensitivity, but on the other hand, it requires large equipment, experienced personnel. It requires that the sample is processed ex situ, and of course, due to the thermocycling and protocols of this technique, you require higher power consumption, which many times is not available in the areas of Africa. So here is where the Diskognosis platform comes to try to fill in the gap and keep the advantages of the mentioned technologies and try to exclude their disadvantages. So the core of this platform is a disk, and called the LabDisk. The handling of the sample is based on centrifugal forces, and this offers the advantage that we need no external pumps, no external tubes, and that increases the automation and the portability of the system. It is like a CD in size, which allows parallelization and multiplexity, as well as integration of the so-called unit operations. That means microfluidic structures, which allow for valving, aliquoting, mixing, and so on, such treatment of liquids. And in addition, it allows the assay transfer from tube to disk. So whatever we do in the lab, it can be miniaturized on a disk. And typically, we can see a figure here, a design with various sections. So in this section, we inject the sample. In that section, in the next one, we have the lysis of the bacteria and binding with magnetic beads. The beads are transferred to the next chambers, washed in order to purify the DNA. Then there's a mixture with the amplification reagents, and finally aliquoting and nucleic acid detection. That is the DNA sector of the disk, if I can call it like this. There is also the protein sector of the disk, where key functions are plasma separation unit, the colored one, here, and then the immunodetection unit. And along with all these, we have special chambers where we pre-store washing buffers in such, aluminum stick packs, as you can see here. So, like a CD, this biodisk needs also a player, like a CD player. This is a figure of such a LabDisk player. These are the dimensions that you can see typically. It's developed by Kiajin Lake Constance company. Some of their capabilities is that, of course, it has a mechanical unit with user-defined protocols for user-defined spinning frequencies, a thermal unit which can be again used, which can use defined protocols for amplification, integrated magnets for handling of magnetic beats, and an optical unit for fluorescence and luminescence. And as they say that one picture is 1,000 words, I believe one video is 1,000,000 words. So I would like to show you briefly a trimmed video of what the device looks like. This is how we typically place the disk. Here's how the disk spins and accurately stops, and we have the detection. And the integrated magnet, which can capture beats from one chamber and move them to the other as the disk moves slowly. And later, you can see the disk, it's reality, okay? And the second short video will show one of those microfluidic functions that I mentioned before. So here you see our sample. What will happen is that the sample will flow into these so-called aliquoting fingers. This is the equivalent of pipetting, what we do in the lab. And then when the frequency increases, this liquid will pass to these reaction chambers, and then the amplification will take place, which is not seen here, of course. So increasing the frequency now, we have the break of these hydrophobic patches and the liquid propulsion. So what do we detect? I was very happy to hear shortly before that, there is a necessity to detect not only the malaria but also other fever-related diseases, and that's why in the project we target malaria, which is parasite, dengue, which is virus, and typhoid pneumonia, which are bacteria. So from the nucleic acid point of view, DNA and RNA, and from the protein point of view, antigens and IgM antibodies. Try to cover as many agents as possible, and to increase our capacity for diagnostic window. So how this multiplexed analysis will take place in very short words, focusing on the protein sector, the amino acids. There will be an integrated chip in the disk, encapsulated in the disk. There will be it will be pre-functionalized with capture antibodies, and the magnetic beads that I mentioned before will act as carriers. These beads will be conjugated with quantum dots, these colors that you see here, and each color will correspond to a particular marker. Let's say that the patient has got disease A and disease D. So in the end, and after washing and after all these procedures taking place all on the disk, we will have a detection of only those beads, those colored, let's call them colored beads, which are conjugated with the marker of the disease. And from the DNA point of view, from the DNA sector, we're going to use the so-called geometric multiplexing, those fingers that you saw before. Each reaction chamber will have pre-stored primers in a dried format, and the quantum dots will be used for quantification, taking signals like this. Of course, using the proper controls and so on. Only a few words about the fabrication technology of these disks. Initially, the first generation was made of rigid materials like PMMA, essentially as rigid as CDs, but then we thought to move on to polymer foils, flexible materials. Main reason was that they help better the thermal conductivity between the small thin-walled chambers. So the amplification technology, the amplification of DNA takes place more efficiently. The process for fabricating those foils is based on thermoforming, and thermoforming is not something new. It's used for pills or blister packages, but it is adapted to microstructures. That's why we call it microthermoforming. On a prototyping scale, it looks like this. So there is a mold, this red shape that you see here. It is placed on this machine. It is heated. The foil is placed on top of that. The chamber is closed, and with elevated temperature and pressure, pushing the foil on the mold, the foil takes the proper structure. And one of the partners is going to undertake the large-scale production in a module-by-module serial line, where we can have the foil starting from a roll and then being structured and being filled with material and finally being sealed and cut and ready for packaging. Instead of conclusions, I thought to include some of the expected impact that we have. From the technological point of view, we aim at finally having a real portable and real point-of-care apparatus, being able to achieve sample preparation in situ, so ideally sample to answer, as we call it. So put the sample, close the cap, press the button, and have the result at the end. That's what's called full automation. And multiplexing, and finally have this installed line batch fabrication technology. The technology of this LabDisk is also adaptable to other applications, for example, other blood-related diseases like neonatal sepsis, which is now running under the ASC Microplat EU project, or using other matrices like sputum for tuberculosis or other respiratory diseases under the project Parzival, again running in our group, or food pathogens, for example, in water, milk, and so on. So, finishing my talk, I would like to thank, of course, the funding agency, the EU, for this 3-year project we started a few months ago, ending end of 2015. I would like to thank our consortium, consisting of 3 SMEs, 3 universities, and 1 non-profit organization, among which is CLINAM and University Hospital Basel. I would like to invite you to visit us at diskprognosis.eu and to thank you very much for your attention.
Thank you very much for the presentation. Questions? Yes, please.
Thank you for the very interesting presentation, first of all. I wonder if there are any limits of this technology.
In terms of what?
For example, cost or some technical issues you are facing that it's not fully developed, or there is space for improvement?
The cost is an important issue because for this particular project we aim at Africa. Therefore, it has to be minimized as much as possible. Knowing from our partners, the main part of the cost comes from the fabrication of the polymer itself, because the components, the reagents and so on, they are down to the order of a couple of euros per test. But adding the cost of the production, currently it would go up to 7-8 euros per test. Yes, but also knowing that there are hundreds of necessary tests to be done in those areas, this means that the technology will have to produce a lot of batches, and therefore price should be lowered.
Just a following of the question, how much is the cost of the production of the machine that you use for centrifugation of the disk holder?
The disk player. But currently it is of the order of a few thousand euros. And with one of our partners, which is the commercializing company, we are trying to see how some sponsoring agent could be possible to contribute to the cost to the countries of interest.
Thank you.
Thank you.
2 questions. The first question is, what is the stability once you've loaded the disk? Shelf life?
So this has to do with the polymer itself, with the pre-stored agents, reagents, and so on. Yeah. At current, and using those stick packs that you saw, a few months, up to 6 months.
And that's in part our task of our group. And here the question is which binding molecule you choose. And if you start with whole antibodies, then you have the downside of the thermal stability. But there are also alternative binding molecules around which might serve as a substitute, which are much more stable. But this is not yet finalized. So this will be something to be optimized during the project duration of this project. And we don't have the results yet, but we are working on it.
Okay. The second part of the question is, have you looked at using really low-cost portable devices instead of this one piece of equipment that you have, like fitting it to a cell phone with a small motor? I mean, this is. I look at it as reducing size and everything else, but still retaining some of the ability.
Yes.
I'm sure you're thinking about it, but.
So the cell phone technology is used by another subgroup of our large group, but this one has to be, as I said, the footprint of SED because it allows the multiplexing and the integration of a lot of structures, and therefore it has to be more or less this size. And the price increases due to the use of a camera for fluorescent imaging. But this, on the other hand, is essential in order to achieve the multiplexing. So, but I would like to add one more technical restriction that's, I guess, somebody would not easily think of. When you need to transfer those disks to Africa with airplane, you really have to take into consideration the pressure difference so that those stick packs, which store the buffers, do not open. So it's a lot of parameters that Mats needs to take into consideration.
I'm sure someone of us will think about this too. Thanks a lot.
Okay. What can also be said is that portable CD players have become very cheap. And the fundamental technology is not so much different. And that means if you think of scale, this will make a huge difference in the price per item. Yeah. But first it needs to work.
Exactly.
Yes?
One more question, please. Are there any errors in detection?
Yes, of course. Well, by saying errors, you mean... Yes, there are, for example, there is, for example, between those aliquoting chambers, there is this coefficient of variation, for example, of the order of 6, 7, 8%. And one problem would be, for example, that we have a gradient of concentration along those chambers. And this is one reason that we try to use one chamber with one chip in order to get rid of these concentration gradients along these chambers.
Just a couple questions. How long does it take to get a diagnosis? That's my one question. Can you reuse this disk, or is it one disk per diagnosis?
It's one disk per test, so it's a disposable.
Okay, and then the third question is, the machine that reads this, what type of electricity? Can you run it off batteries or solar power or something like that?
Okay, that's a good question. Your first question was about the duration. The DNA section, the actual amplification takes place in about 20 minutes. It's not a PCR, it's an isothermal amplification. Together with the sample preparation, I would say roughly it could go up to 1 hour from the point that you inject your sample until you get the result. But again, roughly, because we try to, of course, to minimize as much as possible, we will try to do the best during the next 2.5 years. For the electricity, yes. Now we're using current, so we might be able to use in the post-project era battery or other sources to be fully independent, because I know that there are frequent and long-lasting electricity cuts in those areas, perhaps even no electricity at all. But the fact that we have this isothermal amplification is very good instead of PCR for energy and power efficiency.